Okay, so kind of moving on from diabetes, and we're moving on to different type of disorders now. So now we're looking at the respiratory disorders. But again, just before we get into that, we just need to talk about the basics of gas exchange. So firstly, if we go through the basic laws, we've got Boyle's law. So the pressure of a given mass of an ideal gas is inversely proportional to the volume at a constant temperature. So what that basically means, if you've got a box filled with gas and you decrease the volume, so making it smaller, the particles will start moving around faster, and that will increase the pressure. Similarly, when you breathe in, you're expanding your lungs and therefore decreasing the pressure, allowing the pressure inside your lungs to be lower than the one outside, and the gases will move via a gradient down. So then we've got Dalton's law. So each of the gases we breathe in and contributes to a total pressure in proportion to its relative abundance. So as we know, our atmosphere is made up of different gases. We've got them here, and kind of the main ones, the total is 760 millimeters mercury, and the O2 diffuses across the alveoli and then diffuses cells to endoplasma, and this is because the distance between the capillaries and the alveoli is very small, and also CO2 and O2 are lipid soluble, so they can easily cross the phospholipid bilayer. So then we've got Henry's law. So at a given temperature, amount of gas in solution is proportional to the pressure of that gas. So therefore, if the pressure increases, molecules will go into solution. So if you're thinking about a cell, we've got pressure all around, you obviously have nowhere to go but to go into solution. So if we talk about the structure of hemoglobin and how this oxygen is moved around, so we've got each hemoglobin molecule being made up of 4 polypeptides and 1 heme group associated with each chain. So in the center, we've got this iron Fe2+ molecule in the center that binds with 1 O2 molecule, and each red blood cell has about 280 million hemoglobin. So if we think about how many oxygen molecules that can bind, it's lots. So if you basically remember this graph here, so that's basically the pressure of oxygen at the bottom and the saturation of hemoglobin on the left. So, and this is a sigmoidal shape, and we'll talk about that, why that is later. But yeah, if you basically learn why this graph moves left and right, it tells you basically what you need to do with that. So at low pressure, according to Henry's law, the O2 is unloaded from hemoglobin at the tissues, and at high pressure, such as in the lungs, the O2 is loaded onto the cells. Because when we talk about if there's high pressure, we want to go into solution, so O2 is loaded into the cells. And the factors affecting this and the rate this is done at are these here and the ones in the graph. So if we go through them one by one, so we've got pressure of oxygen. So obviously high pressure of oxygen leads to binding of O2 with hemoglobin, and therefore a leftward shifts, meaning that the less pressure is required to load the oxygen. So then we've got this molecule called 2,3-BPG, and that's the metabolite formed in the red blood cells from intermediate in glycolysis, and the BPG binds to the T-state hemoglobin, so that's the 10th state, and we'll talk about that, and stabilizes it, therefore lowering its affinity for oxygen, meaning more energy is required to bind the oxygen. So when it's present, more oxygen binding sites must be filled in order for transition from the tense to the relaxed state must occur. So at high oxygenated hemoglobin, it inhibits that enzyme that makes 2,3-BPG. And more glycolysis and metabolism means for BPG, which decreases hemoglobin affinity for O2 and leads to more unloading. So in the cells where there's loads of glycolysis and oxygen's being used up, and so we want that oxygen to be unloaded so it can be used for that glycolysis, and carbon dioxide to be loaded on so it can be removed and breathed out. Then we've got the pH and the Bohr effect. So when O2 binds hemoglobin, certain amino acids in a protein reduce H+ ions, so protons, and an increase in these protons will drive the reaction backwards, meaning more O2 is unloaded even at constant pressure. So again, in working muscles, we want this to be unloaded. Also, when H+ binds to hemoglobin, it decreases its affinity of hemoglobin for oxygen. This occurs at high CO2, as it is acidic and turns into carbonic acid with water. So in muscles where this carbon dioxide is produced, the acidity increases and causes again this oxygen to be unloaded onto the cells from hemoglobin so they can keep working. Lastly, we've got temperature, and that alters the shape of the hemoglobin, and as temperature increases, hemoglobin decreases affinity for oxygen. And again, this is why when you're working out and your muscles get warm and you get warm overall, more oxygen is unloaded in the working muscles and more carbon dioxide can be loaded on. Also, when we breathe cold air, it increases hemoglobin affinity for O2 and promote loading. And this is why athletes train in high altitudes. So we've got this, if we now talk about the hemoglobin in more detail and how it works. So we've got this T state, which is tense. So more interactions occur and more stable and has lower affinity for O2. And then we've got this R state, which is relaxed and fewer interactions. It's quite flexible and has high affinity for O2. The hemoglobin has two models, which we go through. And the reason we have two is that none of them explain everything in nature, what happens. So we've got this concerted model, and that states that the hemoglobin can either be in the T state or the R state. So it can be in both states, but just in proportion to each other. So when O2 binds to heme group on the T state, it will shift the equilibrium towards the R state, so there is a more tense than R state. And this occurs as you keep adding O2 and the equilibrium shifts in order to keep the balance between the two. So there's never like an intermediate or anything. And the problem is that this model states that it's either in the T or R state, and it doesn't have the consideration that when one oxygen molecule binds, the polypeptide that it binds to, that it as we see in nature, it changes shape and causes other molecules around it to change shape as well. And this isn't seen in this model, as it can either be tense or in the R state, so it's either one. Whereas in the sequential model, and this occurs when the T state gains an O2 and the conformational change does occur, that limits the concerted model. But however, so we firstly have the T state, and as the O2 is loaded onto, we've got this change, and we go through these intermediates until 4 O2 molecules are bound. And then we have the R state, and obviously as the O2 binds to one of the sites, it changes the other two sites in the hemoglobin molecule, allowing for higher affinity for oxygen in those other polypeptides and allowing the oxygen to bind quicker. However, what this means is that you've got, for example, you've got a T state at one end, then we have 3 intermediates, so with 1 oxygen, 2 oxygens, 3 oxygens, and then you have got 4 oxygens, which is your ready state. However, the limitation of this model is that we see that even if you've got 3 oxygen molecules bound, we see that most of hemoglobin in nature, at least, we see that it's already mostly in the R state, whereas this model only says it's in its intermediate state. So yeah, this is why we've got 2 models, because none of them explain everything. So what happens in the blood when we transport these gases? So if we've got carbon in the blood, so that's 70% carbonic acid, 20% bound to amino acids, and 10% in gas. And the reaction that takes place via this carbonic anhydrase is when we've got CO2 with water, that makes an acid, carbonic acid here. And because this is a weak acid, it partially dissociates into this proton and HCO3-. And this CO2 can then combine with hemoglobin and this amino acid here to form this HBNHCO3, so this carb amino hemoglobin. So what happens when we use up oxygen and the CO2 is used up in the muscles? So what basically happens is that the CO2 diffuses into capillaries and combines with water in the blood to make carbonic acid. So then in the red blood cells, the HCO3 ions diffuse out into the plasma, and because they're negatively charged, we need something to balance out this charge. So the chloride shift occurs to balance out the charge, and entering of chloride ions, which are also Cl-1. Then the HbO2, which is in the oxygenated state, comes along, and that's a strong base, and accepts some H+ ions acting as a buffer. And as we talked about before, the acidity and the Bohr effect causes O2 to be displaced. So O2 goes into tissues which are respiring, and when the CO2 is removed, the O2 goes in. And this is why the graph will then move, the graph that we talked about before, will move to the right. And this can be reversed by high O2 tension in the lungs. So as deoxyhemoglobin, so that's hemoglobin that's bound to carbon dioxide, as we want to get rid of it. So as deoxyhemoglobin bind with oxygen, it releases the H+ ions. And these combine with HCO3- to make carbonic acid again, then into CO2 and water, which are exhaled. So the carbonic anhydrase reaction is kind of reversed. Yeah, so that's why when we breathe out, and when you breathe out on the mirror or a window, you can see condensation occurring because you're breathing out water as well as carbon dioxide. And this increase in PCO2 leads to a decrease in pH again, and lower O2 binding, which offloads this O2, as we mentioned. So we've got this Haldane effect, and that just basically talks about how hemoglobin can carry CO2 in the deoxygenated state better than the oxygenated state. Yeah, that's basically it. So we've gone through the basic laws of gas exchange and how oxygen and carbon dioxide is offloaded. So next lecture we're just going to talk about what happens when things go wrong and we can't properly work our muscles or the reactions aren't taking the right course. So yeah, once again, check in the description for the links to the books, which have been very useful. And if you're interested in buying any questions, and yeah, leave a comment, and yeah, subscribe, and I'll see you in the next video. Cheers.